all for being here today. I'm excited both as the Vice President for Research from Utah State University and as a researcher to share with you the kind of work our students can do in basic research that leads to outputs of potential use in biomedicine. And so today I'm going to be talking about some of our work with a molecule you're all scared of. And so carbon monoxide. But before I go there, I just wanted to say I bring to this the background as a basic researcher, but now as a VPR who thinks about bringing things from our research to market. And so I'm very interested in partnerships resulting from this type of presentation. So please reach out. And so let's start with carbon monoxide and why it might be a potentially interesting molecule to think of from a biomedical perspective. If you leave today with one thought, I want it to be this, that the next time you have a bruise, you look at the colors of that bruise and think about the biochemistry that's going on in the body. It turns out that a bruise represents the stages of the decomposition of heme. Heme is what gives blood its red color, and through the decomposition, particularly in the first step, carbon monoxide is released. The other color changes have to do with the macrocycle that breaks down as the ring opens. But that carbon monoxide release is interesting. And over about 25 to 30 years, various studies, including those involving human participants, have found that low-dose carbon monoxide produces some very interesting biological effects. For example, in cardiac graft rejection, low concentrations of carbon monoxide prolong survival and prevent rejection. There's been a number of studies done around the use of carbon monoxide in organ replacement. In other areas, carbon monoxide can serve as a vasodilator, for example, in addressing pulmonary hypertension. And so this has brought about interest in how to use carbon monoxide. And if one goes into the literature right now, what you will find is that carbon monoxide is actually being explored in low-dose amounts for treating a variety of conditions that vary from things like heart attack and stroke to things that are more bacterial, infections, sepsis, to cancer, and another area that we're particularly interested in, which is inflammation. Over about 20 years, it became apparent that we can't think about using carbon monoxide gas, obviously, because of the high degree of control one would need to have around its use. So researchers began looking for what I will call molecular tools to deliver carbon monoxide. And as I said, over about 2 decades, people have been using molecules that are what I'm showing here. They were molecules that contained carbon monoxide as a unit, but also have other components, such as metals. If one goes to a place like PubMed, you'll find hundreds of citations that are using these particular molecules as what are called carbon monoxide releasing molecules, or CORMs, to deliver CO. Over time, it has become very apparent that they are unreliable in terms of that CO production, that there is independent reactivity going on with the metals that is producing biological effects that have not been considered independently. That inadequate controls were used, particularly to address the possible role of the metals in producing biological effects, and there have been variations from what was seen with CO gas. And so these reliable tools have become problematic and new tools are needed. This is where my group came in 2015 in a metal-free approach. And so we discovered a molecule called a flavanol, which is shown in part A up here. This flavanol structure is light sensitive, and so we can use it to produce carbon monoxide in a light-driven reaction, where the group that's the hydroxyl or OH group in compound 1 is lost as carbon monoxide in a clean reaction. And I show on the bottom the fact that we can do this in a variety of entities. On the left are cells that have been treated with the compound 1. The green channel is labeled, and you can see that compound 1 has a green emission. When we illuminate the sample, that green emission disappears. So the conversion that results in CO loss has a loss of fluorescence. This is really useful because, for example, if one puts it into an entity, as I'm showing on the right, this fish model, what you see is that you can see the green emission, and if we were to illuminate that, the emission would deplete. Another thing I want to mention specifically here, light-driven chemistry for biomedical applications requires that you be able to go to low wavelengths. Our structure 1 can also be triggered with low energy visible light delivered through 2-photon irradiation or other means. And so it means we can do it in tissue, and I'll come back to that. My group at Utah State University has students that range from sophomore organic students all the way up through PhD students. And what they do is they work on the chemistry of this molecular tool now. What they're doing is they're building variations on structure to target particular applications. And so, for example, we can make constructs that will go to specific locations within a cell and deliver CO. We can associate our molecules with protein for protein-related delivery. These approaches allow us to give to the biomedical community different opportunities to use this carbon monoxide releasing motif. Now, I want to be able to show just how this is enabling advancements in the field. And so I'm going to touch on a few other people's work. So after we released our initial papers showing the ease with which you could make our flavanol construct and its reliability as a CO releasing molecule, others have picked it up. And so in this particular example, a group from China picked it up, and you'll notice in red, the flavanol construct has been tied into a long chain and has a blue protecting group on the hydroxyl position. What they're doing is constructing micelles, and these micelles then are able to be used for delivery of this in a biological environment, as I will show you on the next slide. And so this simple first slide shows you that the chemistry that my lab at Utah State University defined holds true here in that the light-driven chemistry first releases the blue protecting group, but then goes on to release carbon monoxide, which is shown emanating from the micelle. And so let's look at how this can be used to look at particular examples that are of relevance for medical applications. And so on the left, I'm simply showing you, as we've talked about before, that you can track these stages with fluorescence. But let's look at the upper right. This is a model of a wound healing. And what you can see is that going across the top is a time sequence from 0 to 12 days, and going down are different treatments that the wound has been exposed to, the wounds have been exposed to, in order to examine different treatments to assist in wound healing. And what this shows is that the second to bottom line is our CO releasing molecule incorporated into a micelle. And what it shows is that the CO releasing effect on the wound is equivalent to that produced from the prior CORM3 molecule. That was one of those metal-based CO releasers. And so this sets the stage to say that our constructs are at least giving the CO effect that has been seen in the past. Looking at a second model, as I said, carbon monoxide has potential uses for a variety of applications. And so the previous one had to do with wound healing. Now I'd like to look at one that has to do with bacterial infection, MRSA. And so let's take a look at, again, they're taking the red framework, that is the framework we've designed and patented at Utah State University, and they've built it into another micelle type molecule. Now this one, and an advantage of our construct, is that they can incorporate other pieces to deliver additional molecules. And so in this case, there's a green piece in the middle. All you need to know is that green construct is delivering a second gaseous molecule, nitric oxide. And so this is going to be a combined CO nitric oxide study. And what we see here is that through light-driven chemistry, these particular micelles can deliver both carbon monoxide and nitric oxide in a co-release. An examination of Staph aureus cell viability as a function of which gas you release or the combo of gases shows you that on the far right, it is the combination of producing both carbon monoxide and nitric oxide that produces the most profound effect. I will note that the carbon monoxide itself is producing a significant effect, but it is the combo that's providing the greatest effect. And so we've seen a number of applications now where the CO releasing motif and its light-driven reaction can be incorporated into micelles, into nanomaterials, and others. At Utah State, what we're doing is we're trying to make the tool better for people so that they can, for other researchers, so that this tool is even more useful. And I'll just mention one recent application that we've published. What we looked at here is by strategically changing the structure to include what's called an amino group, or this nitrogen-containing group on one of the rings, what we can achieve is very low concentration visibility in cells. And so now, using this particular structure, we can see things at just about the nanomolar level in terms of fluorescence. That gives us a way to find out at very, very low physiologically relevant concentrations where the molecule is prior to CO release because of the enhanced visibility. And what we find is that for this particular derivative, we can find it in 3 specific subcellular locations, which happen to be the ER, the mitochondria, and in lysosomes. This allows us to ask questions about if we release CO in a confined organelle, how does it differ from another organelle in terms of the CO release effect? And so we're able to track that, and this paper outlines the initial steps in that study. And so with that, I want to say that my laboratory and my undergraduate and graduate students at Utah State University are continuing to bring new ideas and constructs in this area to the biological community. We are particularly interested in that our molecules are triggered, they're light-driven, they're trackable, their fluorescence gives them a unique property that virtually no other CO-releasing molecule has in terms of the ease with which you can do it. And as I've shown you, they're targetable to subcellular locations. And that targeting work has just begun, because with such a simple organic construct, there's lots of work we can do. And so with that, I want to thank the people who've done this. As I said, my lab at Utah State University has always been an undergraduate-graduate lab, and so I will point out just a couple people. Jason Richards was an undergraduate who was in on the initial discoveries. Stacy Anderson is the co-patent holder with me on the CO release motif, and other students have contributed great insights along the way. And finally, I would like to thank those who have provided funding for us to continue, which have included the National Institutes of Health for a number of years, the American Heart Association, and Utah State University. And with that, I also want to add my collaborator, Abby Benninghoff at Utah State, who is the collaborator who helps oversee the biological studies that we do. And with that, thank you for your attention.